this option to change organisms. But let's look at the kind of comparison we have here right now. So what we have is this chart that has the organism in the left column, an evidence glyph, enzymes and genes for the pathway, and operons. Now, from left to right, what are we dealing with? Okay, organism is obvious. That's whatever organism is in that row. Evidence glyph. The evidence glyph shows a schematic view of the pathway with highlighting for the different reactions in the pathway, indicating why we think those exist. So let's start up here with Homo sapiens. So you can see the very first reaction step here is in magenta. A magenta reaction step is spontaneous or otherwise not catalyzed by an enzyme. And so this can indicate things that are not individual reactions or things that are spontaneous reactions. Then we have green. A green painted reaction indicates that we think an enzyme for that reaction is present and has been identified in that organism. Then down below here, this is a nice little hodgepodge. We have both black, which indicates an enzyme is absent, and orange and green. And we know green indicates an enzyme is present, and orange indicates a unique reaction. Now, how do we have both a black and a green? Well, that's because let's look down at somewhere where this pathway is fully elucidated in E. coli. There are two isozymes that carry out this bottom step. In E. coli, they're both marked as present, and one of them is unique to that pathway. And this uniqueness, why this matters, uniqueness means we've identified this enzyme for this pathway and for no other pathways in this organism. And this might matter because, say, we're showing you a pathway, and it's a predicted pathway, it's one of our Tier 3 databases, and there's only one enzyme we've assigned to that pathway, and it's not unique. In that case, you might look at that pathway and think, maybe I don't believe that exists. But if we assign a single enzyme to that pathway, and it's unique, then you might be more inclined to think that it exists. So in the case of this reaction step in E. coli, we've assigned two enzymes to it. One of them is unique. In the case up here of Homo sapiens, and in the case down here in Francisella, we have assigned one unique enzyme to this reaction step, but we haven't assigned the other isozyme that we might expect to see based on the microbial pathway. Then here we have a list of enzymes that carry out this reaction with their EC number, if that's available, and I can actually click through from this to the object in that specific organism's database. So let's do that now. And here we have this reaction in Homo sapiens. And this is a reaction page, as you should be familiar with if you've played around with the BioCyc database collection or if you've watched the Introduction to BioCyc webinar series. So let's back up. Now, the reaction will be listed, and the reactions will all be there if the pathway is predicted in that organism or assigned in that organism. But over here we have enzymes and genes. And in this case, you notice that two of the enzyme steps have no actual enzyme associated with it. They're just reactions, meaning we don't know of an enzyme that carries out those steps in Homo sapiens. And down below here, in Tularensis, we have a similar issue. To the right here, we have the operons column. In the operons column, we show the genes that code for the enzymes that are involved in the pathway, and we show them in their transcription unit context. So what you can do is, at a glance, compare how regulation works across different organisms. And now we don't have this kind of data right now for Homo sapiens, but we have it for everything else. So for example, you can look at E. coli K12 and you can look at Tularensis, and you can see, well, in Tularensis, FAB-F, FAB-G, FAB-H all together. In coli, FAB-F and FAB-G together. Interesting. And of course, FAB-H is a FAB-F isozyme, so functionally this transcription unit is a lot like this one, and we might want to take a look at the other genes involved in both if we're interested in studying the differences and the similarities between these pathways as they appear in these different organisms. So that's a pathway comparison. Now let's back up a step. Okay, what happens if we look at a pathway that isn't present in all the organisms? Now our drug targetee ideal would be something that's just present in Tularensis, but we don't seem to be seeing that. So how about something that's present in E. coli and Tularensis? like lipid A precursor biosynthesis. So I'll click on that. And we get this. When a pathway is not marked as present in an organism, we're told that. But we still get the evidence glyph, because we might for some reason have a pathway that's not marked as present, but it might have components that are present, and you might want to reevaluate that. Maybe you think you know something that we didn't know when we generated the database, or that the genome annotation folks didn't know when they annotated the genome, and it's useful to you to know that one or more reactions are present. In this case, none of the reactions are present, which is not shocking since this is lipid A biosynthesis. And you can see that the pathways are both completely present in coli and tularensis. So now let's back up to the comparative analysis page. And look at one of the last uses of comparative analysis, which is looking at a single organism. So let's deselect E. coli and Francisella and Homo sapiens. And let's pick something from the Tier 3 collection. Let's go with Nitrosococcus oceani, which is an organism I'm not actually very familiar with. And let's look at everything. So I've selected everything, I've selected my organism, and now I hit submit. Okay, so here's a full report on a single organism. And this is also a good way to show off all the different kinds of things you can see on one of these reporting pages. So again, we have a breakdown of reactions by type. We have reactions of small molecule metabolism, reactions by EC category, distribution of isozymes, the breakdown of pathways by pathway class, a nice long list. And this is a really nice snapshot view of the organism. It's sort of a text snapshot in the way that the cellular overview is a big graphical snapshot of metabolism. And this is nice because you can just at a glance say, well, look, we have 37 amino acid pathways, 33 cofactor pathways, 7 nucleoside and nucleotide pathways, no aldehyde pathways, no chlorinated compound pathways. Is that interesting? Is that special? Well, it's something to look at. Pathway holes. Then we have this pathway hole analysis. And so pathway holes are reactions in pathways that we've identified or predicted in that organism that don't have an enzyme assigned to them. And this represents opportunities for research, or opportunities for you to curate an organism and bring it even closer to the reality of the experimental data for that organism. And while our process involves both predicting pathways and then predicting enzymes to fill pathway holes, we will still have pathway holes left at the end of the day when we make a Tier 3 pathway genome database. So, for example, we know that in this organism we have 227 pathway holes, which is 29% of the total number of reactions in pathways. We have 100 pathways without holes. We have 54 pathways with a single hole, 34 with missing 2 enzymes, 8 that are missing 3, 7 that are missing 4, 2 that are missing 5, and 9 that are missing more than 5, and 114 total pathways with holes. And so this is interesting, and actually if we were to click on one of these links, you'd see a list of pathways with 1 hole. And you can click on these individually. Let's go with alanine biosynthesis. And you see alanine biosynthesis, and actually, because this is a Tier 3 database, if you go down here, you see the pathway evidence glyph. Even when you're not actually doing a comparison, that evidence glyph is there, and it will explain to you on what basis we've predicted that pathway in that organism. So let's back up.